Welcome to the Explainer. Today we're doing a deep dive into Corynebacterium diphtheria. Now this is a pathogen with a pretty fearsome history, but it's still very much a threat we need to understand. All the information we're about to cover comes from a Gemini deep search, and if you want to see all the source material, you can check out the full notebook.
So here's our roadmap. We're going to start with the bug itself, look at its toxin, then move into the lab for diagnosis, cover the clinical treatment, and finish up with the big picture public health response. Okay, first things first, let's get to know the organism we're dealing with. What does Corynebacterium diphtheria actually look like and what makes it tick?
So when you get this on a gram stain, there are some classic tells. It's a gram-positive rot, of course, but it's what we call pleomorphic, meaning it's not uniform. You'll see these characteristic club shapes. But the real giveaway, the thing everyone remembers, is the arrangement. Because of how it divides, this kind of snapping motion, it forms these V shapes and L shapes that look like Chinese letters. You might also see these intracellular metachromic granules, which are basically phosphate storage units. It's a very distinctive picture.
Now, from a taxonomy standpoint, this is where it gets really interesting. It's in the macrolide group, which, as you know, also includes heavy hitters like Mycobacterium. And what that means is that it has mycolic acids in its cell wall. Think of it like a waxy raincoat. This makes it super resilient and really helps it survive out in the environment.
The species gets broken down into 4 biotypes. And you've probably heard of gravis and metis, which sound like they mean severe and mild. Well, historically, that's what people thought, but we now know that's not really true. Any biotype that has the toxin gene can be deadly. So today, biotyping is pretty much just an epidemiological tool for tracking outbreaks.
All right, so we've met the organism, but what makes it so dangerous? Well, it's not the bacterium itself, really. It's the weapon it carries, the diphtheria toxin. This is what turns a colonization into a potentially fatal disease.
And here's the fascinating part. Corynebacterium diphtheria isn't born bad. It has to acquire its weapon. A strain only becomes toxigenic after it gets infected by a specific virus, a bacteriophage that's carrying the tox gene. The virus injects its DNA, it integrates into the bacterial genome, and boom, the bacterium is now armed. We call that lysogenic conversion.
The mechanism of this toxin is just brutally efficient. It's a classic AB toxin. The B subunit is like a key. It finds the lock on our host cells, especially heart and nerve cells, and gets the whole toxin inside. Once inside, the acidic environment releases the A subunit, and its only job is to find and destroy one single target, elongation factor 2. This completely shuts down all protein synthesis, and the cell just dies. Get this, a single molecule of that A subunit is enough to kill a cell. Incredible.
Now, depending on where this bug sets up shop, you get 2 wildly different diseases. You have respiratory diphtheria, the one we all think of with the pseudomembrane, the bullneck, and a huge risk of the toxin going systemic. But then you have cutaneous diphtheria, which is more of a chronic, punched-out ulcer on the skin. And because the immune response in the skin is so strong, systemic complications are actually pretty rare.
And here it is, the hallmark of respiratory diphtheria, that pathognomonic pseudomembrane. This thing isn't just a film of pus, it's a tough, leathery mat of dead cells, fibrin, and bacteria. And the key clinical sign, the thing that separates it from, say, strep throat, is that it's stuck on tight. If you try to scrape it off, it bleeds.
So what happens when that toxin breaks loose and gets into the bloodstream? It goes straight for high-value targets. Myocarditis is the big one. It's the leading cause of death. And then there's this really sneaky delayed complication, polyneuritis. Weeks after the initial infection, the toxin's damage to the myelin sheath and nerves can cause paralysis. The kidneys can take a hit too.
Okay, the stakes are obviously incredibly high. So how do we in the lab definitively nail the diagnosis? It's a really important cascade of tests that has to balance speed with accuracy.
The workflow starts with a good sample. You've got to get that swab under the edge of the pseudomembrane. Then it's off to culture on special media like Loeffler's or Tinsdale agar. For a fast result, we run a PCR looking for the tox gene. But, and this is a huge but, a positive PCR is not the end of the story. The gold standard, the final confirmation, is the Elek test, which actually proves the bug is making active toxin.
And this brings us to a really tricky problem in the modern lab, the NTTB dilemma. These are non-toxigenic tox gene-bearing strains, so they have the gene, and your PCR is screaming positive. Public health is getting ready to deploy. But the gene is broken, so they can't actually make the toxin. This is exactly why you can't stop at the PCR. You have to do the Elek test to separate a true threat from a genetic ghost.
For those of us still working the bench, especially if MALDI-TOF isn't an option, the biochemicals are still king for species ID. And with Corynebacterium diphtheria, it's often about what it doesn't do. The key differentiators are that it's urease negative and pyrazinamidose negative. Those two negative results are your ticket to separating it from other common corniforms.
All right, let's say there's a suspected case, the clock is ticking, you absolutely cannot wait for the lab to call back with a confirmation. The clinical response has to be immediate and aggressive.
Now let's talk antimicrobial susceptibility. If you look at the latest EUCAST breakpoints, they tell a really interesting story. Just look at the breakpoint for erythromycin. An MIC of just over 0.06 is considered resistant. That is an incredibly low number. It tells you that the wild-type organism is supposed to be exquisitely sensitive, which really underscores the importance of macrolides. And that brings us to this, a really big deal that just happened in 2024. The World Health Organization officially updated its guidelines. Because of documented rising resistance rates around the world, penicillin is no longer the recommended first-line agent. Macrolides, like azithromycin, are now the go-to treatment. But you know, global guidelines are one thing. It's fascinating to see how they're adapted locally. For example, while the WHO recommends azithromycin, if you look at the Dutch national guidelines, they actually prefer clarithromycin. It's a perfect reminder that you always have to check your local protocols and antibiograms.
Now, what about for kids? The principles are the same, but the details are critical. Antibiotics obviously are dosed by weight. But here's the key thing. The diphtheria antitoxin dose is based on the severity of the disease, not the child's weight. And of course, with laryngeal diphtheria in a small child, airway management is absolutely paramount.
Okay, let's pull back from the bedside and the lab bench for a minute and look at the big picture. Because even a single case of diphtheria triggers a massive public health response. So what does that defense look like?
In the hospital, infection control is strict and it depends on the site of infection. Respiratory diphtheria means droplet precautions. The cutaneous form means contact precautions. And getting a patient out of isolation is a high bar. You need 2 consecutive negative cultures taken at least 24 hours apart, and this is after their antibiotic course is completely finished.
And this brings us to the central and kind of frustrating paradox of diphtheria control. Our vaccine is a toxoid vaccine. It's fantastic at teaching your body to neutralize the toxin so you don't get sick. But here's what it doesn't do. It doesn't reliably stop you from becoming an asymptomatic carrier. And that is why even one case is a public health emergency. A fully vaccinated person can still potentially carry and transmit the bacteria.
So this leaves us with a really critical question for the future. As our molecular tools get more and more sensitive, is our biggest challenge going to be sorting out the real threats from these non-toxigenic gene-bearing mimics? It's a huge debate for labs and public health. What do you think? Drop your thoughts in the comments below. And if you found this useful, please go ahead and like and subscribe. Thanks for joining this explainer.